Hi everyone. Up until now, we have primarily addressed the 16S rRNA gene in high throughput sequencing. Although the 16S rRNA gene has been widely used to study bacterial communities, we need to remember that our microbiomes are far more complex. In part 3.5 of Module 3, which will also be my last lecture for this course, we will briefly be looking into other components of our microbiomes and ways to study them, so I hope you enjoy. As I mentioned as part of Module 2 of the course, more than half of our body's total cell count come from our microscopic colonists. And yes, of course, these colonists do not only belong to the kingdom bacteria. Bacteria, and hence the 16S rRNA gene, only represents one component of our microscopic colonists. In addition, there are also, for example, archaea, viruses, and fungi inhabiting our bodies. One way of studying our entire microbiome is via shotgun sequencing, also referred to as metagenomics. The term multibiome has now even been used to holistically refer to the biodiverse collection of bacteria, viruses, fungi, and multicellular helminthic worms colonizing the mammalian intestine. This review, for example, discussed new insights into the multibiome-host interactions in the context of host protective immunity and immune-mediated diseases. So we've discussed the role of bacteria in health and disease during the course of these modules. But when it comes to the virome, which have been estimated to consist of 10-fold more particles than bacterial microbes, understanding its impact on health and disease is in its infancy. Here are just a few examples of more recent papers highlighting the importance of the gut virome in health and disease. Although the microbiome, which is the fungal constituent of the multibiome or microbiome, is less diverse and abundant than the bacterium, fungi have been shown to activate the type 17 axis of the immune system and can contribute to gastric and systemic diseases. Some of these previously reported include gastric ulcers, food allergy sensitization, and colitis, as well as allergic airway disease. This slide just highlights how studies have recently started to focus on the microbiome. And then there's the intestinal multicellular parasitic worms, most commonly referred to as helminths. The role of helminths in immune responses have been studied widely. Unlike bacteria, fungi, and viruses, helminths are only present in about 1/3 of the global population. So how do we study the multibiome? Well, one way of doing this is to use shotgun sequencing. But how does it work? Compared to 16S rRNA gene sequencing, both methods require nucleic acid extraction from the sample. During 16S sequencing, we amplify the region of interest and sequence only that region. Hence, we only obtain information regarding bacteria. Another limitation is that we don't necessarily obtain enough resolution to perform species-level analysis when only targeting a single or few regions of the 16S rRNA gene. During shotgun sequencing, the entire genomic content is fragmented and segments are sequenced. Genome consensus is then constructed computationally. But despite the fact that we can identify organisms in kingdoms in addition to bacteria, and we are able to investigate functional pathways, it is very expensive to conduct shotgun sequencing and computationally intensive. So just to recap, in summary, the microbiome does not only refer to our bacterial communities. In addition to bacteria, our bodies are also inhabited by other microscopic and sometimes macroscopic organisms such as archaea, viruses, fungi, and multicellular parasitic worms. The collective term used for these organisms is the multibiome. Although studies focusing on the virome and microbiome are still new, reports have been published on the importance of these organisms in health and disease states. One way of studying our microbiomes as a whole is to use shotgun sequencing. However, using this technology is still very expensive and computationally intensive. So that's the end of Module 3, focusing on the implementation of 16S rRNA gene sequencing when studying the bacterial communities. I really hope you guys enjoyed.